DNA is a type of nucleic acid, which is a form of organic molecules. Its function is to store and transmit genetic information in the cell. DNA stands for deoxyribonucleic acid, and it is made up of nucleotides that contain a deoxyribose sugar, a phosphate group, and a nitrogenous base. There are 4 different bases, and they form complementary base pairs. So adenine pairs with thymine and cytosine pairs with guanine. Also, the bases can be joined in any order. DNA has a double helix structure with a sugar-phosphate backbone and bases that form rungs in the middle. The nucleotides are linked together by covalent bonds, and the 2 DNA strands are held together by hydrogen bonds between the pairs of complementary bases. DNA replication is a process that occurs in the nucleus where a cell doubles its DNA before cell division. First, topoisomerase unwinds the double helix, and then an enzyme called DNA helicase unzips the strands of DNA by breaking the hydrogen bonds. Then single-stranded binding proteins stabilize the separated DNA strands. On the leading strand, the DNA polymerase, another enzyme, adds nucleotides one at a time onto the 3' and using the base pairing rules, creating an identical copy of the DNA that has one old strand and one new strand. Since the strands of DNA are anti-parallel, the leading strand is easier and can be synthesized continuously, while the lagging strand is synthesized in fragments that are then joined together. On the lagging strand, DNA primus adds RNA primer, which the DNA polymerase can bind to and use as starting points to replicate the DNA. So the DNA polymerase will add nucleotides and then move back to add some more, and then move back again, repeating the process. This forms disjointed segments of the new DNA, which are called Okazaki fragments. Finally, DNA ligase binds the fragments together to complete the DNA replication process. RNA, or ribonucleic acid, is a molecule that helps turn genetic instructions into proteins. It's similar to DNA, but it has a few key differences. RNA is single-stranded. It uses sugar ribose instead of deoxyribose, and it has the base uracil instead of thymine. There are 3 main types of RNA, and each one has a special job in the process of making proteins. Messenger RNA is made in the nucleus using DNA as a template. It carries the instructions from DNA to the ribosome where proteins are made. Think of it like a photocopy of a recipe being sent from the DNA to the cell's kitchen. Transfer RNA brings the correct amino acids to the ribosome. Each tRNA has an anticodon that matches with the codons of the mRNA. It acts like a delivery truck that brings the right ingredients for the recipe. Ribosomal RNA helps form the structure of the ribosome and helps with reading the mRNA. It's kind of like the kitchen counter where all the protein making happens. Together, these 3 types of RNA work like a team to make proteins the cell needs to survive. Transcription is the first step in making a protein. It happens inside the nucleus of a cell. During transcription, the cell makes a copy of the gene's instructions in the form of messenger RNA. First, an enzyme called RNA polymerase finds the gene on the DNA strand that needs to be copied. RNA polymerase then unzips a small section of the DNA. It uses one side of the DNA strand as a template and then builds a single strand of mRNA by matching the RNA bases to the DNA bases. Adenine pairs with uracil, not thymine like in DNA. Cytosine pairs with guanine. This process continues until the whole gene has been copied. Once the mRNA strand is complete, it detaches from the DNA and leaves the nucleus, heading to the ribosome in the cytoplasm. Now the instructions are ready to be translated into a protein. Translation is the process of building a protein, one amino acid at a time, from the genes in the DNA. All 3 types of RNA are used in the process, and it occurs on a ribosome in the cytoplasm of a cell. The first step is initiation. First, the RNA in the ribosome binds to the mRNA at the start codon. Then, a tRNA molecule carrying the designated amino acid, which is coded for by the codon on the mRNA, joins onto the mRNA on the ribosome by bonding the codon on the mRNA with the anticodon on the tRNA. Then the large ribosomal subunit joins the complex, forming the complete ribosome for translation. Next, in the elongation stage, the ribosome moves along the mRNA strand and reads the codons, which code for a specific amino acid that is brought in by the corresponding tRNA. As the tRNAs connect to the mRNAs, the amino acids that they are carrying form peptide bonds and create a polypeptide chain. Finally, in the termination stage, after releasing the amino acids, the tRNA strand leaves, and once a stop codon is reached, the polypeptide chain is released from the ribosome, marking the end of translation since the RNA has been successfully translated into a chain of amino acids which forms a protein.